Hello, I'm Dr. Tydell again, and today we're going to start talking about molecular biology. And by the time we're done with this particular chapter, we will also have discussed quite a few enzymes, all right? So before we start, though, I wanted to tell you this, that feeling of frustration that you feel when you're trying to learn or understand a new concept is the feeling of your brain making new connections. Let's do two concepts. Concept number one, you already learned all of the easy stuff. So you may look back on elementary school or high school and think, oh, it wasn't that hard to learn back then, right? Probably not, because that's all the easy stuff. You got the easy stuff out of the way. The rest of this stuff that's in college, yeah, well, it's harder. And it's not anything that you should expect to peruse and have it instantly in your brain. All right. These are concepts you need to wrestle with. Not just my class, but pretty much all the classes you take now that you're in college. So that's number one. Number two, sometimes when people feel really frustrated and they're like wrestling with the concept and they're so confused, they think it means that they're lost and doomed, that they're not going to learn a concept. That's not what it means at all. Let me give you an analogy. Let's imagine that you've got a friend who's never worked out at a gym before. And so you bring them to the gym with you, and together you are doing biceps curls to make pretty biceps, okay? And they start to do the biceps curls just like you just did. And they're like, ow, this hurts. And they set the bar down. And then you do your set and they do their set. And hey, it hurts again. This isn't working. I'm leaving. There's no point. I'm never going to get in shape. What would you say to them? You'd say to them, hey, dude, that burning feeling in your muscle, that doesn't mean it's not working. That means it is working, right? Same thing for you. When you're confused and you're frustrated with yourself and you're like, why is this so hard? I want you to just remind yourself that's the way it feels when your brain is busy making those extra synapse connections that we will learn about in this class. That's the way it feels, okay? It means it's working. Now, that being said, back when I was in school, our only option was really to read the book again and again and again and again, look at your notes, or if you were really gutsy, try to go and talk to the professor. First of all, those are all your options. Remember my Zoom office hours, you can just drop in, okay? But you have got so many more options. Go to other things on YouTube, right? You're watching this, you're probably on YouTube. So you can go to Amoeba Sisters, Amoeba, Amoeba Sisters. They're really good on this particular topic. Or I've been posting crash course videos, right? Or there's also a Professor Fink, Fink, or you can find the same concept at Khan Academy, Khan Academy, all one word, .org, right? Get other people to describe it, and you will find those aha moments where you will go, oh, and you'll say to yourself, why didn't Tydell just say that? And maybe that's what I should have said, or maybe your brain's working on it, working on it, working on it. And that last thing you watch just was that final puzzle piece that made the whole thing make sense. Okay? So don't give up. So protein structure, let me remind you, because we already talked about protein structure, that proteins and DNA, nucleic acids, and even carbohydrates, they are real three-dimensional things. Yes, they're microscopically small, but they're not ghosts, okay? They're actually real, and they have shape, and we learned the levels of protein structure, right? Primary structure was the sequence of the monomers. The monomers of proteins and peptides are amino acids, right? Secondary structure are things like the alpha helix, the beta pleated sheet. I said the secondary structure was the analogous to wheels or a windshield, right? Tertiary structure is ultimately what's the shape of the protein. Quaternary structure is for big proteins like hair. There's zillions of these little guys being put together in quaternary structure. So This is what's important. The amino acid sequence is going to dictate the secondary structure, which is going to dictate the tertiary structure, etc. And we know that ribosomes build these proteins. So how do the ribosomes know which protein they're supposed to make and what are the instructions for making it? That's molecular biology. Here's something you should know, pretty sure it's on exam 1, that that the DNA that's inside of the nucleus, the reason it's there is to tell your ribosomes which proteins to make and to give them the recipe for how to make them. Okay? So the DNA is divided up into areas that are called genes, and every gene that you have is a recipe or a blueprint or an instruction guide for how to build a protein. That's what genes are. Okay? So here's your nucleus right here. This is a chromosome once it's condensed. By the way, most of the time, your DNA does not look like that, okay? But we're used to seeing it like that. And your DNA is in the form of chromatin. And if we unwind it, it looks like this double helix. And you already know that DNA is a nucleic acid, and the monomers of DNA are nucleotides. All right? Now, this whole story of molecular biology has got a lot of detail, right? Before I jump into all of the detail, I want to start with one of my analogies. Yeah, by the way, if you don't like analogies, I'm sorry. I just think in analogies, and so I teach in analogies. But we're going to start with this analogy, a 2-part mold, okay? Now, let's just imagine that you're an archaeologist and you have found at some wonderful dig site this piece of ancient pottery shard, and you happen to find the ancient mold that piece of pottery came from. Ooh, okay. Now look at how pretty they look on that little piece of board. You decide, I am going to be rich. I am going to make hundreds of these. I'm going to sell each one of them as a piece of ancient pottery artwork. I'm going to sell them for $1,000 each, and I'm going to get rich. Okay. Now, you are the architect who found the archaeologist who found these. How are you going to do that? How are you going to make thousands of these so that you can sell them? Are you going to hire an artist to carve that and then hire another artist to carve that? No. What you would do is you would hire people to put clay on the outside of this piece of pottery, and when it dries, it'll form that thing. And then you'll hire someone else to put clay on this piece of pottery, and when it dries and you put it in an oven or whatever, it would be that piece, right? The two pieces fit together, and by using this piece here on the left, you could make perfect whatever that is on the right. And by using this piece on the right, you could have perfect whatever that is on the left, right? That is the way DNA is. DNA is double-stranded. Let's see, I'm going to try using a pen. Okay, here's one strand, right? That is one strand, and I'm going to use a different color for the other strand. Blue, I don't know. Okay, here's the other strand. Okay, they are two strands, and one of the strands is this guy right here, and one of the strands is that guy right there. Okay, what do I mean by that? So see on this piece of pottery, everywhere that there is a straight ridge in the piece of pottery, there is a straight groove in the mold. And everywhere there is a round ridge on the pottery, there is a round groove in the mold. And you're like, okay, yeah, I understand the way molds work, Tidal. Yeah, but that's the way DNA works. And now you might think, sure, okay, whatever. Do you know we didn't discover this until the late 1960s? Now that seems like forever ago to you, but it was recent enough that when I was in college, they didn't teach me this because they didn't really know it yet. It hadn't made it into college classes yet. It took us forever to figure this out. Now, is it exactly made out of clay? No, it's not made out of clay. They're molecules. However, wherever there is a C, we'll get back to what the C is, on one side, there's a G on the other. That's like a rounded ridge with a rounded groove, right? And wherever there's a straight ridge on one side, there's a straight groove on the other side, and vice versa, right? The two-part molds. Now, why? Well, why do this? Okay. Well, I just asked you, if you wanted to make thousands of these guys, would you hire an artist? No, you wouldn't, because if you hired an artist to carve them, it'd be expensive and the artist would make mistakes, right? If you want identical, whatever this thing is on the right, the best way to make it identical is to put clay on this thing on the left. DNA needs to be copied millions of times in your lifetime. Millions, actually, probably more than millions. Yeah, probably hundreds of millions. So if there were mistakes, even 1 in a million times, you would end up with cancer and you'd be dead. Okay? You can't have mistakes when the DNA gets copied. This whole system that we're going to discover, we're not going to discover it. We're going to discuss it. This entire system we're going to discuss. The point is going to be how to make copies of this information without making mistakes. And the system that has been developed is just like a two-part mold, okay? We are going to start there at the next video.